In the next three chapters, we're going to cover some biochemistry. And the way we've broken this down is that we're going to spend each of these chapters looking at the three macronutrients: carbohydrates, proteins, and lipids. So let's start off with carbohydrates. So carbohydrates are they have a wide variety of uses in biochemistry. I think one of their more fundamental uses is, especially as sugars, just energy storage molecules. They're very, way to store energy that's readily accessible for most organisms. In the case of some of the more complex carbohydrates, they can be used for just for structural purposes. So think like cellulose in plants. And of course, going down to things like DNA and RNA, the sort of rungs of our double helix in DNA come from that carbohydrate polymer forming that. Now, the term carbohydrate implies that you've got carbon and water. That's not entirely true. You don't have literal. It's not the same setup as you see in a hydrate salt, where you have water molecules embedded in the crystal structure. Instead, here the water comes from alcohol groups and hydrogens being attached to each carbon in your carbon chain, save the two terminal ones. So basically, I'll show you a Fisher projection of this in the next slide, but you can see here that if you look at each of these carbons in the middle, we have our carbon with an alcohol group and a hydrogen that's not shown. The H and the OH forming the water of the quote-unquote hydrate part of that. Okay, though even though technically that's not a hydrate. You probably remember from high school biology or just lower level biology in general that glucose, one of the fundamental carbohydrates, fundamental sugars, is produced by plants by photosynthesis. So plants, green plants, take carbon dioxide and water vapor, and in the presence of sunlight and chlorophyll, they convert that into glucose. All right, and we get oxygen as a byproduct. And of course, this glucose works its way up throughout the food chain, right? You've got plants that eat animals that eat plants, animals that eat other animals, and so on. Okay, so this is basically a way to fundamentally take sunlight and have it sort of power an ecosystem. Okay, now a quick note I want to draw your attention to. Notice that each of these carbons here are chiral inside our chain, right? We've got basically chiral centers where we've got four different groups coming off of those carbons, right? Our alcohol group, the hydrogens that are not shown, and then this carbon group and this carbon group that are different from each other. Also note that the two terminal carbons are not chiral. We have two hydrogens over here, so that's just a CH2 group. And over here we have a carbonyl. Okay, so that's going to be something that we're going to notice in all of our carbohydrates. There's going to be a carbonyl present at one end of the molecule. In this case, it happens to be an aldehyde, but though that need not be the case. You could have a carbonyl on that second carbon, then you'd have a ketone, but we'll talk about that in a little bit. Okay, so here's a Fisher projection. We talked about Fisher projections way back in the first semester when we were just talking about how to represent molecules in general. And Fisher projections are very convenient here because of all those chiral centers in the middle of our molecule. Okay, so the thing that's really convenient here with your Fisher projection, based on the setup that we've got here, is that the alcohol group kind of tells you what the chirality or what the designation is for each of those chiral centers. Basically, if your alcohol group is pointing to the right, that's an R. Basically that is R chirality. And if we have the alcohol group on the left, it's going to have an S designation. Okay, so you can name these accordingly. So you've probably heard a few names of carbohydrates at this point. I've probably said things like glucose and cellulose, and you've probably heard of other carbohydrates just from general knowledge, things like fructose or lactose or sucrose and so on. So you've probably noticed a pattern there, right? That ose ending at the end. that basically is going to be the general suffix for a carbohydrate. So you should be able to readily recognize it. I'm going to add a few more terms here. We're going to make this, distinctions between aldoses and ketoses. And we're going to then have other names that have a Greek prefix for a number in front. So things like pentose or a hexose or a tetros and so on. Okay, so what do those two things tell us? Well, you've probably started to guess. The difference between an aldose and a ketose is the presence of an aldehyde group or a ketone group. So you can see over here glucose is an aldose. Okay, our carbonyl is on that number 1 carbon, and therefore that's an aldehyde. Okay, that's what makes that an aldose. Fructose, on the other hand, has its carbonyl at the number 2 position. Okay, so that carbonyl has 2 carbons attached to it and therefore is a ketone, and that makes fructose a ketose. Now, for the designations like pentose and hexose and so on, you have to count the number of carbons in your chain. So here we've got 1, 2, 3, 4, 5, 6. So glucose is a hexose with its 6 carbons. Fructose has 1, 2, 3, 4, 5, 6 and is also a hexose. All right. So the smallest monosaccharide we can have with 1 chiral center is glyceraldehyde. All right. So it's a triose, if you will. You've got 3 carbons here. Okay. Now, you can have at that 1 chiral carbon, you can have your alcohol group pointing right or pointing left. So, again, if our alcohol group is pointing to the right, it's that R designation, and if it goes to the left, it's an S designation, right? However, notice that we also distinguish between our 2 isomers, or our 2 enantiomers of glyceraldehyde using this plus or minus designation. Now, if you recall, basically when you have, when we look at light shining on our, plain polarized light shining on our sample of our enantiomer, if we have just 1 enantiomer, it'll deflect it one way, and its enantiomer, the other enantiomer, will deflect it the other way, right? And that's where we have these plus or minus designations, or dextrorotatory or levorotatory designations, right? D or L. So in this case, the isomer, the enantiomer that has that R designation happens to be dextrorotatory, all right? So you'd have a D here or a plus here, and this chiral center happens to be R, right, recto. Whereas here, the one where that chiral center is to the left, sinister, happens to be levorotatory. Okay, so this obviously begs the question then: can we predict, based on the RS designation of our chiral centers, whether our monosaccharide is going to be dextrorotatory or levorotatory? And the answer is no. You cannot. You have to determine that experimentally. This is, by the way, a very common trick question. You can pretty much guarantee that you'll see it on a quiz or exam, or both. So watch out for that. You might remember this trick question from back in that chapter when we learned about doing this experiment to determine whether things are dextrorotatory or levorotatory, right? You have to. That R and S designation tells you about the structure itself, but that does not necessarily tell you how it's going to deflect the light. Now, when we look at other sugars, we use that last chiral carbon, the one at the bottom, furthest away from the carbonyl, to designate that D or L designation, right? So if we have our alcohol group pointing to the right, so it's got an R configuration, we say that that sugar is a D sugar. So so here we have D ribose, D glucose, and D fructose, respectively. But now you've probably noticed a contradiction in what I just said, right? We're using that D designation there, even though we don't know. You need experimentation to figure out whether this will deflect light to the left or to the right. Okay, so what this means then is that you can't, that D doesn't refer to dextrorotatory anymore. Okay, you have to use plus or minus designation for dextrorotatory and levorotatory. D just here just refers to that sugar's configuration, kind of like how we refer to R and S, except we use R and S for individual chiral centers. Here we use R and S, we use D and L for effectively the entire structure, but it's determined from that bottom carbon, okay, that bottom chiral carbon. Okay, so for example here, both of these molecules are D sugars, right? If you look at that bottom chiral carbon, they have that alcohol group pointing to the right, and they're both aldotetroses, right? They have an aldehyde group and they have 4 carbons, right? So they're tetros. But they are different sugars, right? Because they notice that they differ on that top chiral carbon. Okay, so again, you could call these erythros and threose, but in order to, distinguish them from their enantiomers, you would have to call them, D-erythros, because that alcohol group's pointing to the right, or D-threose, because it's pointing to the right. Okay, so that tells us then that there are 4 aldotetroses. We just went over 2 of them. Okay, you've got D-erythros and D-threose. So what are the other 2? Well, as I pointed out, you've got the enantiomers of these, right? You can have L-erythros and L-threose, where you just have the mirror image of these. So L-erythros has, both of its alcohol groups on the left. Okay, and L-threose has its alcohol groups on the right and the left, respectively. Both of these chiral centers are flipped. Okay, so there we go. So that's what that looks like. Okay, and again, if you ever get, just shown a structure like this and you're like, well, what's the designation for it? You can always look at that bottom carbon and, see if that alcohol group's pointing to the left or the right, and that'll tell you if it's D or L. Okay, in terms of whether you've got erythros versus threose, I guess the fact that these are both the same, okay, it tells you that they're erythros, and the fact that they're opposites tells you that these are threose. Okay, quick note here about your sugars. Don't stress too much about memorizing the configurations of the structures of all of the individual sugars. On a quiz or exam, I'll provide you with structures if you need it, or I'll give you some information. I don't expect you to memorize, that the chiral centers in erythros in D-erythros have to be R and R. That's not something I would expect you to know. Okay. All right. So next up, we have the aldopentoses. So we just went over the aldotetroses. We've got aldopentoses here. So we have aldehyde groups at the end and 5 carbons. Okay, and now we have 3 chiral centers instead of 2. Now, here we're going to have 8 different isomers that we can find. 4 D isomers, and then their 4 enantiomers. Okay, so notice that we went from 2 isomers of glyceraldehyde, the 2 enantiomers, right, plus or minus glyceraldehyde, because it had 1 chiral center. And then we went from that to 4 isomers of tetroses, which had 2 chiral centers. And now we've got 8. So notice that we essentially are getting a number of isomers using the formula 2 to the power of n, where n is the number of chiral centers. Okay. So again, this hopefully should be ringing a bell from from back when we first learned about chiral centers way back in chapter 5. All right, so here are the, 1/2 of all of those for the aldopentoses. So we have our 3 chiral centers. We have ribose, where all of these are on the same side. And here we've got arabinose, xylose, and lysose. And of course, if we just do the mirror images of each of these, we then have the L isomers. Okay. Again, some of those names are probably, probably sound a little familiar. Ribose, of course, is the R in RNA, okay, and deoxyribose, where you basically remove one of these alcohol groups and replace it with a hydrogen. That's basically the deoxyribose. That's the D in DNA. Okay, xylose may also ring a bell. You may remember that from along with cellulose as being part of the sort of the building blocks of wood. Okay, So if we scale up to hexoses, you could probably figure out that instead of 8 isomers, there've got to be 16, right? 2 to the power of 4 would be 16. And out of those, 8 are the D isomers and 8 are the L isomers. Okay, so we've been talking about aldoses for quite a bit. Let's talk a little bit more about ketoses. I mentioned earlier that in the case of a ketose, your carbonyl is at the number 2 carbon in your carbon chain of your carbohydrate, as opposed to the number 1 carbon that would make it an aldose. So having it at that number 2 carbon then means that we have, it's got 2 carbons coming off of it, of course, which makes it a ketone group. Now, what this then does is it removes the possibility for another chiral center. So, for example, in the case of a trialdose, your central carbon is chiral, so you can have 2 variants there. In the case of of a triketose, there is no chiral carbon. Notice our number 1 carbon has 2 hydrogens on it, making it achiral, and our number 3 carbon also has 2 hydrogens on it, making it achiral. And of course, our carbonyl is trigonal planar and therefore achiral anyway. All right, so you can see here that for our triketose, there's only one possibility in dihydroxyacetone. Okay, so it's when we move up to a tetros here, a tetraketose, that we have our first introduction of a chiral center and the possibility of a D and an L isomer. So you can see here as we insert more and more carbons, we extend our carbon chain, we get more chiral centers and increase our number of possibilities. So once we get to 5 carbons, once we get to pentoses here, now we have 2 chiral centers, which gives us 2 to the power of 2 or 4 different isomers. Okay, so here are the 2 D isomers, and of course we have the corresponding mirror images which aren't shown. Then once we get, insert another carbon and we have 8 possible isomers, and again here are the 4 D isomers shown. And that's about it in terms of the more common ketoses you're going to come across. Again, don't stress about memorizing the chiral configuration, the RNS configurations of every possible sugar. For the most part, you'll be given a reference in a quiz or exam. Okay, so let's talk a little bit about how, so far we've seen these Fisher projections that show our monosaccharides in a sort of a straight line, a straight chain, but in solution, these monosaccharides actually take a cyclical form. So let's talk about how that happens. The mechanism for this is actually something you've seen before in our chapter on aldehydes and ketones. It's actually the formation of a hemiacetal that we see, because keep in mind that your carbohydrates have alcohol groups and carbonyl, so you already have the pieces you need to form a hemiacetal. So what happens is your alcohol group, one of your alcohol groups, acts as a nucleophile and attacks that carbonyl carbon, and basically converts that carbonyl oxygen into another alcohol group, and in doing so closes the ring. Okay, now typically you'll get 5 or 6 membered rings. They're just more stable from a torsional strain standpoint. So that's typically what you're going to see. All right. Okay. So I'm going to gloss over this. We've talked about hemiacetal formation before. Like I said, basically you have your alcohol group do a nucleophilic attack here, and these electrons swing up here, and then you've got a negative charge, and that just gets protonated. Okay, so in the case of a monosaccharide, though, the important thing is, how do we draw that cyclic product, and how do we maintain or represent the chirality of all the other carbons, right? The configurations of all of them. Okay, so when we draw that closed, that cyclic formation, the cyclic structure, we usually draw it in this form, what's called a Haworth projection. So we have our 5 or 6 membered ring here, and we represent all of our other carbons, the groups coming off all of our other carbons, using these straight lines pointing up or down. Okay, and essentially all that's different, really, you could use this template and just draw that, and then take your Fisher projection, and based on your R or S configuration at each of those chiral carbons, just place your alcohol and hydrogens accordingly, pointing up or down. Okay, so we'll talk about how to do that, but you can see here that essentially when we do our Hayworth projection, our number one carbon is this one over here at the right. Okay, and you can see that we no longer have a carbonyl. This becomes an alcohol group. Now, whether that alcohol group's pointing up or down, we'll get into that. That's the alpha or beta versions of this, or the different anomers, so to speak, of this Hayworth projection. I'll get to that in a second. For each of these other carbons, look at your RRS configuration. It might help to think of your basically you're rotating this clockwise. So this carbonyl carbon is coming down here and to the right, as shown over here. And accordingly, all of your groups, any groups that are to the right are pointing downwards. So you can see here, for example, in this number 2 carbon, this alcohol group's pointing to the right. It's pointing downwards over here. And likewise, any groups that are on the left are pointing upwards. So you can see here on this number 3 carbon, we have our alcohol group to the left. Over here in our Hayworth projection, it's pointing upwards. Okay? All right. In the case of our number 6 carbon, since it's the alcohol group on our number 5 carbon that closes the ring, that's our oxygen over here. Obviously, there's not going to be any groups coming off of that. That oxygen's happy with its 2 bonds. That 6th carbon comes off of our 5th carbon here. It's pointing upwards like that. I mentioned earlier that the alcohol group on that number 1 carbon can point either up or down. Now, the reason for this is that when your monosaccharide is in its open form, your carbonyl is flat. Remember that sp2 carbon is trigonal planar. So that alcohol group can attack either from the top or the bottom, and accordingly you'll get either one of these anomers. Okay, so anomer, by the way, spelled A-N-O-M-E-R, and that's the difference between these two molecules when you form that alcohol group. So if it attacks from the top, the alcohol group forms pointing downwards, and vice versa. If it attacks from the bottom, the alcohol group points upwards. Okay, so if that alcohol group is trans to your CH2OH group here that's pointing upwards, okay, we call that alpha, the alpha anomer. And if they are cis, if those two groups are cis, that number 6 carbon and that alcohol group are cis to each other, that's the beta anomer. Okay. Sometimes it might be easier. Usually, since this number 6 carbon is usually drawn pointed upwards, it's typically that alcohol group's downwards for alpha and upwards for beta. That's another quick way usually to recognize that. Quick note here about the ring-closed names of our glucose rings once it forms that hemiacetal. Typically, depending on if you have a 5 or 6 membered ring, your suffix here will be either furanose or pyranose. The reason being that furan is a 5-membered heterocycle where you have an oxygen present. Think tetrahydrofuran, the solvent, okay? And pyran is a similar molecule but with a 6-membered ring. So you have a 6-membered ring like this, but with an oxygen as one of the heteroatom making up that ring. Okay, so in other words, if you swap out all these alcohol groups and this CH2OH group for hydrogens, you would basically have pyran. Okay, so that's where the name comes from. In terms of the relationship between your alpha and beta anomers, note that this number 1 carbon is now a chiral center, right? So while it was achiral as a carbonyl, once that alcohol group forms, these are 2 chiral centers. So accordingly, keep in mind that all these other chiral centers are staying intact. You're only flipping that anomeric carbon, and so these 2 molecules are diastereomers of each other. Okay, so Yeah, here we have our two anomers. You can help distinguish between them. I mean, obviously from the structure by looking at whether the alcohol group is cis or trans to that number 6 carbon. But also experimentally, you can, since these are both diastereomers of each other, they'll deflect light differently. Now, if I didn't mention it earlier, these cyclic hemiastiles are the stable form of your monosaccharides in solution. If you didn't notice on an earlier slide, I basically mentioned that we have a very, very, very small percentage of the open form, and instead these are more stable in solution. Now, there is a slight difference between the stability of these two, but the fact remains that you get a mixture of these two in solution. So something to keep in mind that even if you start off with a pure anomer, you'll wind up having one converting into the other, and you'll get a mixture of the two. All right. Now, that process of forming a mixture of these two anomers is known as mutarotation. Okay, so basically, you've got if you start off with one anomer, it'll go via the open form as an intermediate, but it will eventually reform a cyclic hemiastyle. And when it does that, it might go back to the original form, or it might produce some of the other anomer. And so you get a mixture. Now, again, you might have one form dominate over the other. You'll get a higher percentage of one versus the other, and that's fine, but you're going to get a mixture of the two. Okay. Now, another thing to keep in mind with the way we draw these things, typically with sugars, we'll draw them as either these Fisher projections or these Haworth projections. They're usually the simplest, easiest way to see where our alcohol groups are on our chiral centers. That being said, keep in mind that these Haworth projections are basically a 6-membered ring, and as such, we can draw a chair form just as we would for cyclohexane. Okay, so basically you might see a Haworth projection instead of drawn as a Haworth projection. You could draw it instead as this chair form. You'll sometimes see this chair form drawn, especially in the case of disaccharides or polysaccharides, since this takes up a little bit less space than our big Haworth projection, and kind of shows what this looks like in 3D. This is sometimes a better way to draw it when showing it in the grand scheme of a larger disaccharide or polysaccharide. All right, so you'll probably notice that later on. But like I said, if we're looking at the stereochemistry of the individual chiral centers, our Fisher or Haworth projection is usually more useful. Okay, let's practice this. So here we have a Fisher projection of D mannose. Let's go ahead and draw the Haworth projection of this. When you draw the Haworth projection, draw the pyranose. So draw a 6-membered ring with the oxygen sort of in that top right corner. I guess we haven't really seen any examples of furanoses yet, but anyway, draw a 6-membered ring, a pyranose of D mannose. Okay, so if you've paused this and come back, I'll go ahead and put up the slide. But again, remember, you want to flip this 90 degrees to the right, okay, or flip it clockwise. And you're going to have basically all of your groups that are on the right pointing downwards, all the groups that are on the left are pointing upwards. Please note the question is asking you to draw the alpha form. So your alcohol group on your number 1 carbon, your anomeric carbon, is going to be pointing downwards, as you can see here. So here's that anomeric carbon. Okay, so there's your Haworth projection of that, and that's good practice. Also, I guess drawing the chair conformation is not a bad practice either. But you can see once you've got you can use your chair template. Just use this oxygen here is that atom here, right? You can use that point of your chair over there. It's basically if you have a group pointing downwards, it's kind of very similar to how we drew cyclohexane chair conformations before, right? If you have a group pointing downwards on a down carbon, it's axial. Likewise, if it's upwards on an up carbon, it's also axial. So you can see that in the case of these two axial groups here. Okay, if you have an up group on a down carbon, it's equatorial, and vice versa. A down group and an up carbon is also equatorial. Okay, so keep that in mind. So if you have this oxygen as an up atom, have this carbon is down, this one is up, down, okay, in your chair. All right, and then accordingly, decide whether your groups are going to be axial or equatorial from that. Okay, so here are so here we have a ketose. Ketoses can form both furanoses and pyranoses. Okay, so this is what a furanose ring looks like, by the way. Okay, so here's our 5-membered ring. We have that oxygen at the top of the pentagon that's formed. And so, with a pyranose, we've seen that the oxygen's at the top right of the hexagon that we draw. Okay, but here's a furanose. And yeah, in the case of our ketose, we can have either our alcohol on our number 5 carbon closing the ring, okay, to make our furanose, or we can have our alcohol on our number 6 carbon closing the ring, and that makes a pyranose accordingly. Okay, so please note that at that point, the number 6 carbon has only 2 hydrogens on it, and so both your up and down groups are hydrogens there. I mentioned earlier with our examples of aldoses that the open chain, the open form of our monosaccharide is typically not very stable in solution. We can see that again here with our ketose. We have a very small percentage of that. But please note that we have all the different forms of our ketose present in solution. We're going to get different percentages of those. So we're talking about the alpha and beta anomers of both the pyranose and the furanose. Okay. Now, while the beta pyranose is the most the highest percentage that we see here in the case of fructose, it's actually the furanose ring that we see taking part in most of the reactions in biochemical pathways. Speaking of which, let's talk about the reactions that monosaccharides can undergo. So generally, your functional groups are alcohol groups and carbonyl. So those are the main branches of reactions that we're going to look at. One key one is to swap out our alcohol groups for something that's not protic. So remember, the alcohol groups engage in hydrogen bonding, and that's what makes your monosaccharides water soluble. So if you want to improve your solubility in an organic solvent, you need to swap out those alcohol groups for something that's not protic. So one way to do that is to take acetic anhydride and pyridine, and that will replace your alcohol groups with acetate groups. All right. The pyridine is used to soak up the extra protons there. Okay. Another functional group we can convert our alcohols into is ethers. So instead of esters, we convert them into ethers, and this is easy enough by taking easily accomplished by taking a alkyl halide and having your alcohol come in as a nucleophile. So it basically does an SN2 substitution and swaps out those alkyl groups, or swaps out the protons for alkyl groups. So you've got the corresponding ethers. I mentioned that our cyclic form of our monosaccharides is a hemiacetal, right? So in order to close that ring, your alcohol group, converts your carbonyl into an alcohol group, and therefore you've got your hemiacetal. If you react your hemiacetal with an alcohol, you'll convert it into an acetal. We've seen this back in our chapter on aldehydes and ketones. And that's no different here. All right, so we can swap out that anomeric alcohol group for an alkoxy group by reacting with an alcohol in the presence of an acid. Please note that the acetal equivalent of our sugars is known as a glycoside. That word's going to sound familiar later on when we talk about glycosidic linkages. Another thing to keep in mind is that since you go through an sp2 hybridized intermediate, the resulting product, you get a mixture of both the alpha and beta anomers of your acetal. Okay, it doesn't matter which anomer you start off with, you wind up getting a mixture of those two products. Okay. Another really important thing to note here is unlike the previous examples where we did reactions at all of the alcohol groups, here it's only the anomeric alcohol group that's replaced. All right, and so here's the mechanism. Again, this should look pretty familiar from what you remember of acetal formation from hemiacetals. Another interesting reaction we can see here is the conversion between glucose and mannose. Now, glucose and mannose are a special type of diastereomer called epimers. So epimers are basically diastereomers that only differ at one position, okay, at one chiral center. Whereas, diastereomers can, if you have multiple chiral centers, diastereomers can include all of the combinations. Epimers are only if there's one chiral center that changes. So basically all epimers are diastereomers, not all diastereomers are epimers. Anyway, the number 2 carbon here is the one that's relevant. Okay, so what basically happens is under strongly basic conditions, you go through this diol, an ene diol. You've got an alkene with 2 alcohol groups. So you've got this intermediate here. And you can, of course, go back to the original monosaccharide. But it's possible then instead to get, when that carbonyl reforms, it's possible to have the stereochemistry of that chiral center inverted. Okay, so notice that we've gone from an sp2 carbon, so from an sp3 carbon here to an sp2 carbon here, and then back to an sp3 carbon. Again, we have a racemic mixture. So my point here is that you can change the sugar itself just by having a strong base present. Next, let's look at oxidations and reductions. The carbonyl can be reduced to an alcohol group. So in this case, we can take the aldehyde group in glucose and reduce it to a primary alcohol here using a strong reducing agent like sodium borohydride. Okay, so when we do that, the suffix changes from -ose to -itol. Okay, so you've gone from an aldose into an alditol. Right, and so that's also known as a sugar alcohol, and it's basically this is one type of artificial sweetener. Okay. We could go the opposite direction. We could take an aldehyde and oxidize it to a carboxylic acid. So if we use bromine as our oxidizing agent, it will only oxidize that aldehyde group. Okay, and so we only form the carboxylic acid here. Please note that none of the other alcohols are oxidized to ketones or anything like that. Okay, so just bromine forms a resulting acid. We call this an aldonic acid. All right, so we take that ose ending and change it to onic acid. Okay, so please note that we only oxidize our aldehydes here, right? We can't, if we did this, use bromine as our oxidizing agent, which I've just pointed out is mild enough not to, oxidize anything other than the aldehyde group, ketoses won't be oxidized by something that mild. So you have to use something stronger if you want to oxidize a ketose. Okay, so this includes using certain metal ion agents such as silver ions or copper 2 ions, and those will oxidize both aldoses and ketoses. The way they oxidize ketoses is they actually tautomerize them into the ketone group into an aldehyde group, and then the aldehyde group can then be oxidized. All right, so any sugar that can react with one of these oxidizing agents. So usually in the form of, in the case of silver ions, it's usually in what we call Tollen's reagent, or in the case of copper 2+ ions, that's usually in the form of either Benedict's reagent or Fehling's reagent. So you've probably heard of those tests for carbohydrates or for reducing sugars. Those sugars that give a positive test here are known as reducing sugars. They're so called because they reduce the metal ions into the metals, right? While the aldehyde group or resulting aldehyde group gets oxidized into a carboxylic acid. Okay, so again, this is only at that carbonyl position. Okay, if you want to oxidize anything other than that, you need to use something stronger as your oxidizing agent. So in this case here, we're going to use nitric acid. If we use nitric acid as our oxidizing, as our oxidizing agent, not only will we oxidize the aldehyde group at one end into a carboxylic acid, but we will oxidize that terminal alcohol group on our number 6 carbon into a carboxylic acid as well. Okay, so notice that now we have two carboxylic acids at both ends. So this is no longer this is not just a gluconic acid, but this is a glucaric acid. Okay, so again, the generic term here is an aldaric acid. Okay, you just take the ose ending and change it to aric acid. Now let's look at some reactions that we can, use with synthetic applications. So one of these reactions is to use hydrocyanic acid, which will react with sugars to make cyanohydrins. Basically, at what used to be your carbonyl, well, you convert your carbonyl into an alcohol group, and then you have a nitrile group present at that same carbon. Okay, so it's kind of like a hemiacetal, but instead of an alkoxy group, you've got a nitrile group. Okay, now instead of basically, you can take this nitrile group and convert it into a carboxylic acid, and then reduce it into into an aldehyde group. Okay, so once you've done that, notice that it's kind of like the original starting molecule, but you've inserted a carbon with a hydrogen and an OH group on it. Okay, so in other words, you've extended your sugar's carbon chain by one more chiral carbon. For example, if you started off with a pentose, you would have converted into a hexose. So that was one version of the Kiliani-Fischer synthesis. The more recent version, which is a little bit more effective, it has higher yields because instead of going via a carboxylic acid intermediate, you're going directly to the aldehyde. Basically, you can use hydrogen and a poisoned catalyst, so palladium with barium sulfate, and it reduces the your nitrile into the corresponding imine, which you can then hydrolyze into directly into an aldehyde. Okay, so so basically that's so keep that combination of reagents in mind for the Kiliani-Fischer synthesis. In other words, if you have a sugar and you want to extend it by one chiral carbon, reacted first with hydrocyanic acid, and then subsequently reacted with hydrogen, lead and barium sulfate as a poisoned catalyst, and subsequently hydrolyzed it with water. Okay, so please note, of course, though, that you get a racemic mixture here. You get both versions of that chiral carbon. Okay, another synthetic application that we can apply, to carbohydrates is kind of the reverse. Basically, you can take a monosaccharide and you can take out a chiral carbon, take out one of the carbons in its carbon chain. So in this case here we have glucose. We're effectively taking out the number 2 carbon here, and we're going to turn it into arabinose, which is a pentose. We're going from a hexose to a pentose. So what we do here is we take hydroxylamine and react it with our sugar. It converts this our carbonyl into this corresponding sort of hydroxyl imine, all right, also known as an oxime. All right. We then react that with acetic anhydride, which then converts that into the corresponding cyanohydrin. So we take that group and we're converting into a nitrile group. Okay, and of course that carbon it's connected to has an alcohol group, so that's why this is technically a cyanohydrin. You then react it with an alkoxy group, which basically deprotonates that alcohol and kicks off your nitrile leaving group to reform an aldehyde group. Okay, so just like we started off with an aldose here, you wind up with an aldose in the final step. Okay, now what you've done then is you've kind of you've basically taken off your carbonyl group at the end, your aldehyde group at the end, and you've converted the number 2 carbon into the new aldehyde group. Okay, so far we've been talking about monosaccharides, but I've mentioned that monosaccharides are essentially monomers. We can link together monosaccharides to form chains of these sugars, basically, disaccharides and polysaccharides and so on. So a disaccharide is when 2 monosaccharides connect together, and they connect together through a glycosidic linkage. Now, I mentioned earlier that a glycoside is the acetal version of the ring form, the cyclic acetal, hemiacetal version of your monosaccharide. In that, we took an alcohol and reacted it with our hemiacetal to convert into the acetal. Well, here our alcohol is another monosaccharide, right? Remember, monosaccharides have alcohols all over them, so one of those is the alcohol that converts your hemiacetal into an acetal. Okay, so how these monosaccharides can link together, which alcohol groups are involved, that's what makes different types of disaccharides. And, when we get further, you'll see it makes different types of polysaccharides as well. Okay, so it's the difference in these glycosidic linkages that gives us different types of carbohydrates. Okay, a very common one you're going to see is a 1,4 glycosidic linkage. So what does that mean? Well, remember your anomeric carbon is the one that's where that used to be your carbonyl, and that's where your hemiacetal and your acetal forms, right? It's always that carbonyl carbon. So that's your number 1 carbon. It's the number 4 carbon on the incoming monosaccharide, that nucleophile, that causes that glycosidic linkage. Okay, so that's why it's called a 1,4 glycosidic linkage. Now, here we have maltose. Maltose is a common example of a disaccharide where we have 2 glucose molecules linked together through this 1,4 linkage, specifically through a 1,4 alpha glycosidic linkage. Okay, so what that means is that the alcohol group, that anomeric carbon, right, that alcohol group coming off our anomeric carbon in the original electrophilic monosaccharide, that was an alpha anomer. That alcohol group was pointing downwards. Okay, so you can kind of see that even in these 2 monosaccharides linked together, that the the oxygen is pointing downwards. So you can tell that that used to be your anomeric OH group, and it's in the alpha position. Okay. So cellobiose is kind of like maltose. The only difference here is you can see that this is a beta glycoside. So it's also a 1,4 linkage, right? It's still that number 4 carbon on the other glucose molecule coming in. But the difference is, of course, that now this oxygen's pointing upwards. Okay, so it's in the beta position. Oh, quick note here about the ability of disaccharides to be reducing sugars. Even though the anomeric carbon of the glucose on the left is used up, so this anomeric carbon isn't able to convert back into an aldehyde, and therefore it can't be oxidized further. Please note that the alcohol group here over here can be opened up. This anomeric carbon can be freed up and converted back into a carbonyl, back into an aldehyde group, which can then, be oxidized into a carboxylic acid. Okay, and that's the case for both maltose and cellobiose. Both of these are reducing sugars because this anomeric carbon here and its alcohol group can be converted into an aldehyde if you open up this ring. Okay. Here we have lactose. Lactose is a disaccharide formed by galactose and glucose. So unlike cellobiose, where you have 2 molecules of glucose linked together by a 1,4 beta glycosidic linkage, here we've swapped out the our monosaccharide on the left in our electrophilic monosaccharide into we've swapped that out with galactose instead of glucose. Okay. Oh, so by the way, you've probably heard of lactose intolerance. The reason for this is that people who have trouble digesting lactose have trouble breaking. They don't have the necessary enzymes to help break down this linkage to get the galactose and glucose out of lactose. So in other words, it stays lactose. So your body can't. People with lactose intolerance are unable to digest this into its monosaccharides. Okay, sucrose, also known as table sugar, is another disaccharide. Here we have glucose connected to fructose. All right, notice that fructose here is in its beta furanose form, and yeah, basically it is a 1,2 glycosidic linkage. All right, now in the case of a 1,2 linkage here, where in the number 2 carbon is the anomeric carbon on our ketose here. All right, so remember, fructose is a ketose, and this used to be a carbonyl carbon when it's in the open form. Notice that neither of these monosaccharides are able to have their carbonyls reform, right? You can't open this up and reform a carbonyl because that oxygen is being used up in forming this glycosidic linkage, right? So therefore, sucrose is not a reducing sugar. Okay, so it seemed probably like a lot of these examples we've gone through were reducing sugars. Here's an example of something that's not a reducing sugar. So this would not react with silver ions in Tollen's reagent or copper ions, copper 2 plus ions in Benedict's reagent or Filling's reagent. And so you would get a negative test with those. Okay, so if we can link together two monosaccharides to form disaccharides, you can keep doing this and form what are known as polysaccharides, which are basically polymers of monosaccharides. Okay, so a very common example of this is cellulose. So cellulose, it has a 1,4 linkage between glucose molecules. So it's basically kind of like cellobiose, but just a continuous chain of it. Okay. Different kinds of starches are other examples of polysaccharides. They are also, so for example, like amylose over here is also made up of glucose, but unlike cellulose, where you have a 1,4 beta linkage, here we have a 1,4 alpha linkage. Right. Amylopectin is another example of this. Unlike amylose, amylopectin is branched. Basically it's like amylose, but in addition to just having your 1,4 alpha glycosidic linkages forming the main sort of chains of your polymer, it branches off at the number 6 carbon. Okay, so that alcohol group coming off your number 6 carbon can be the nucleophile connecting an anomeric carbon on another branch. So in other words, you have a 1,6 alpha linkage. Another variation of these sugars is to swap out an alcohol group for an amine group. So when we do that, we call those amino sugars. So for example, here we have glucosamine, which just has one of these alcohol groups swapped out for an amino group. All right. Now, if you take a glucosamine and swap, basically acetylate it, all right. So basically, we form amides here. This corresponding polymer chain of that is chitin. All right. So chitin, you may have heard of before, is the kind of skeletal armor on a lot of invertebrates. So things like crustaceans and insects have their skeletons are made, or their shells are made up of chitin. Now, in the example we just looked at with glucosamine, we swapped out that number 2 alcohol group for an amino group. In the case of N-glycosides, it's the anomeric carbon that has an amine attached to it. Okay, so you can see here if we take a monosaccharide here and react it with a primary amine, we get the corresponding N-glycoside, and that amino group is tacked on here. All right. Again, notice that even if you start off with only one anomer, you get a mixture of the two different anomers here. All right, so again, it's going to go through an sp2 hybridized intermediate. All right. Now, if you've heard the term N-glycoside before, or have it ring a bell, you're probably thinking of it from RNA and DNA. Okay, so the N-glycosides in those are called nucleosides. All right, so for example, here we have a ribonucleoside where we've got the ribose. If this were an OH group, this would be the sugar ribose. So we've swapped out that OH group for an amine. Okay, so now of course, depending on what this base is, you've got different types of ribonucleosides, right? And that's where you can enter codes essentially into your RNA, or in the case of deoxyribonucleosides, notice it's deoxy because this alcohol group's missing. That's the deoxy part. Here in the case of DNA, by changing the base present here and making a polymer of these of the sugar parts of these nucleosides, we can essentially sort of put in a sort of chemical code by just varying those different bases. Okay, so of course, and so in the case of both RNA and DNA, there's 4 different bases we can use: cytosine, thymine, adenine, and guanine. You've probably seen those abbreviated just by their initial letters, C, T, A, and G. Okay, and so just changing the combination of those bases, the sequence at which those bases show up on your polymer chain, is how DNA can store, genetic information. Okay. In the case of your DNA, so we had nucleosides earlier without the phosphate group. When you tack on a phosphate group, this becomes a nucleotide. Okay, and this is actually how your different. These are each of the monomers in your DNA chain, and it's basically these phosphate groups that connect to the alcohol group on another nucleotide to make a phosphoester. Here you can see how each of our nucleotides are linked together to form sort of the backbone of your DNA strand. Okay, so this is basically how you form that phosphoester. Notice that the phosphate group on one sugar, one ribose, connects to the alcohol group on another ribose. Okay, now basically DNA forms complementary strands. Basically, cytosine and guanine can hydrogen bond efficiently, and adenine and thymine can also efficiently hydrogen bond with each other. So that allows these 2 strands of DNA to sort of link together. Okay, and so that's why when you see DNA and you see that double helix pattern, actually I think there's a picture on the next slide, the hydrogen bonding between the different bases sort of forms like the rungs of a ladder, okay, and cause the that phosphoester backbone to sort of twist around. They're kind of like the uprights of the ladder, but instead of being like a vertical ladder, it's kind of almost like a spiral ladder or a spiral staircase. Okay, and so that's basically how 2 complementary strands of DNA can wind together. All right, this we're not going to go into the biochemistry here, but basically when DNA replicates, it takes advantage of this fact, because when you unzip a strand of DNA, you essentially can use that favorable hydrogen bonding interaction to sort of recreate the complementary strand, and that's how you convert one strand of DNA into 2 strands of DNA. Okay, and that's how DNA replicates itself. Okay. I mentioned that RNA also uses four bases. The difference is thymine. Thymine is not found on RNA. You instead find uracil. Okay, so that's the one difference. But otherwise, all the others are the same: cytosine, adenine, and guanine. All right. Yeah, and they pair up very similarly as well. Okay, so again, here you can see like the strand with RNA accordingly over here. All right. Now the difference with RNA is that you don't get that double helix like you see in DNA. All right. But the fact that you can form complementary strands is again how RNA is able to replicate itself. All right. And that's it for this chapter. So yeah, let's go over some additional practice problems. All right, so this question's a bit of an essay one, so I'm just going to gloss over that. You're probably not going to see something like that on your quiz or exam. I think this is another essay one, so let's skip over this one. Okay, here's something that might be a little bit more applicable. So here we have, so take a sugar, take, like let's say glucose. I would start off with its in its cyclic hemiacetal form, and ask yourself how would it react with an amine, okay, and what would it make? For your amine, you could just use a generic amine, so just have like let's say a primary amine with an R group on it, just the generic R group, okay. All right, so if you've taken a stab at this, hopefully you would have realized that your amine coming in as your nucleophile is going to attack that anomeric carbon, right? So here's the mechanism for that. Here we have our, basically our glucose, or sorry, our monosaccharide, and we can protonate that the alcohol group on that anomeric carbon, and it's a make it a better leaving group, and so we can make this sp2 hybridized intermediate, right? And so that anomeric carbon is now easier for more prone to an attack from our nucleophile. So that a lone pair from our from our amine can attack either side of this. So here we have it attacking from the top, it looks like, but we could easily have it attack from the bottom. And when it does that, it forms this protonated intermediate, which you can then, you have an amine, you have lots of amine floating around, which is a base. You can deprotonate that and give us the neutral, N-glycoside. Okay. Which of the molecules discussed in this chapter could be actually classified as polymers? There's a lot of those, but so if you've paused this and thought about it, this includes things like you can just have polysaccharides like cellulose, amylose, amylopectin, and all of those have glucose as the monomer. So again, remember it's the glycosidic linkage, the type of glycosidic linkage that changes the type of polysaccharide. Okay, I mean, of course, you can change the monomers as well, but the fact that you have different types of glycosidic linkages yields you greater diversity in terms of polysaccharides you can make. All right. In terms of non-glycosidic linkages, non-polysaccharide polymers, we have things like chitin, which remember was a glucosamine converted into an amide. So I guess glucosamide, but anyway, a polymer of those. All right, and then of course we had DNA and RNA, where you had each of your monomer units linked together. Basically you'd have a phosphate on one monomer linked together via the alcohol group and another to make a phosphoester bond. Okay. So that's it for the chapter. These, like you probably noticed, those practice problems weren't super helpful. So do take a look at some of the practice problems suggested in the homework, and yeah, if you have any questions about what's coming up on the what the quiz is going to cover, just feel free to ask. But again, it'll be more of the kind of application type questions. So definitely like, go over the synthetic problems we looked at, a lot of the synthetic applications, a lot of the reactions of monosaccharides. That's going to be useful. We're going to have some declarative things. So like, be able to like look at the structure of a monosaccharide and be able to classify it accordingly. Like say like, oh, this is an aldose and this is a tetrose or whatever, right? Be able to identify glycosidic linkages, stuff like that. To be able to like, classify things, be able to do application type problems involving reactions, and so on. Okay, and as I said, if you have any questions on that, just let me know.